Is it okay if I move this camera? Because it's like staying this way. I don't know if I should touch it. Don't, don't touch it. Oh, yeah. No, it's now it's clocked in on Tyler over there, which is fine. Tyler's beautiful, but like if it should be.
Just come on
Oh,
over
it
here.
likes you.
It likes
Yes,
me. Sit
Kyle.
right over here. Sit right over here. Yeah, it knows I own it.
Okay.
There. Hold on.
Okay. All right. Do you think? Yep. Cool. All right. Thanks for coming to Wednesday morning, 8:30. My name is Kyle Clonin. I'm with the New Jersey Water Supply Authority. We've worked a lot with USGS, and that's how I know Pam Riley. If you want to introduce yourself, Pam.
Sure. Happy to be here virtually. My name is Pam Riley. I work for the New York Water Quality Center, Water Science Center. I am the Integrated Water Quality Section Supervisor. Nice to see you all virtually.
So many water quality managers use continuous monitors and remote sensors nowadays to identify and manage harmful algal blooms. A lot of the remote sensing helps with saving time going out to the field and assessing a full water body, and the continuous sensors are great for showing live data. And as this continues to get integrated, it starts, leading to management. And we are very excited to have this panel here of government and private sector experts to discuss these tools, the progress they're making, and how they use them to make decisions in management for harmful algal blooms. Our panelists include today Bridget Seegers of NASA, who works on the Cyan project, Reagan Herrera of the NOAA Great Lakes Environmental Research Laboratory, Matt Titus of Clearwater Analytica, Tyler Arnold from Veolia North America, Lauren Weinrich of American Water, and Jessica Ciceli of Ohio Water Microbiology Laboratory for USGS. So with that, what we're going to do is have each of the 6 panelists show a quick slideshow, introduce themselves and their work. We might have time for 1 or 2 questions there, hoping that takes about 10 minutes each. That'll be an hour, and then a half hour for kind of group discussion questions. And we'll go in between both people online, which Pam will manage, and people in person. I'll be walking around with a microphone later. So our first speaker who's going to come up is Bridget Seeger. Dr. Seegers is a research scientist and NASA lead on the Cyanobacteria Assessment Network, or CYAN project, that provides satellite-based cyanobacteria estimates for water bodies across the US. Her research often combines in situ data with satellite remote sensing to better understand regional dynamics and events such as harmful algal blooms. Before returning to graduate school, she taught for 6 years in a range of non-traditional classroom settings, from the woods of Minnesota to Boston and at a charter school in San Diego. Dr. Seegers is passionate about her outreach and education, and one example of this is a really cool sounding grant project she received to bring the NASA Earth Science and the CYAN project to people at restaurants, bars, and breweries, primarily in small towns in the lake regions of Wisconsin and Minnesota. The event was called Sippin' on Science and engaged hands-on science activities, trivia, and informal presentation on lakes, algae, research robots, and satellite science, with the goal to break down boundaries and create opportunities for the public to ask questions and talk to a scientist. Dr. Seegers is an avid surfer and sailor, and thank you for being here.
Great. Good morning. It's great to be here. So, yep, I'm Bridget Seegers. I'm a research scientist over at NASA Goddard. I do a lot of freshwater work, but I'm housed in the Ocean Ecology Lab. It's really nice to be here among all you freshwater enthusiasts. And also it's nice to be here because I grew up in Wisconsin, so it's really great to be back where I fell in love with water and also cheese. So it's nice to be sharing that with you as well.
Oh, one thing, we can't see your presentation, Bridget, yet. While we have this break, I just want to remind our folks online, for all of our virtual participants, that if you have questions for our speakers, please put them in the Q&A in Whova or in Zoom, not in the chat. And please do not raise your hand. We're not monitoring that. Thanks. Take it away, Bridget.
So as was mentioned, I'm going to be talking today about a project I've been working on called CYAN, which is short for the Cyanobacteria Assessment Network. This is an interagency project with the EPA, NOAA, USGS, and with us at NASA. And the goal here was to create the ability to do satellite remote sensing of cyanobacteria blooms across the United States. And we did it, which is great. So if you don't know a lot about cyanobacteria, they are an issue because they can cause HABs, a harmful algal bloom. They create toxins, which has impacts on human health and recreation. So a big health issue as long in an economic issue as well. So our standard product for CYAN looks like this. We create the true color images and then also the cyanobacteria index. So you can look, you can see the bright colors. That's an indication of cyanobacteria in the lake. If you see gray, that means the level of cyanobacteria is below detection. And any black in the image means there's no data. So typically that's either because of clouds or maybe a lake might be too small to get a nice image for. This data comes from the Sentinel-3 satellites. There's a sensor on each of those satellites called ULCHI. It has a 300-meter resolution, and we have near daily coverage. So we get about 6 images a week. So not quite daily, but near daily. And this morning, you can get yesterday's image. So there's about a 24-hour lag on the images that you're going to get. And now we were supposed to give some examples. So one of our users is the Department of Environmental Quality in Utah. They had a sampling regime of about every 2 weeks to 10 days to go out and do routine sampling. This is an image from Utah Lake. This is June 25th. They had actually just done sampling 2 days prior. So they weren't scheduled to go out sampling again until after the 4th of July holiday. They started looking at the data and they noticed this cyanobacteria bloom had kicked up. So they sent a team out, took samples, and sure enough, there was toxins in the water. So they started closing beaches. Oops. The image on July 3rd looked like this. So now they have a very widespread cyanobacteria event. Again, they had already closed a lot of beaches. This is a huge recreational lake, and they prevented a lot of people from getting exposure. Resources for the Future did an economic study on this single event, and they estimated that $368,000 were saved in societal costs because of better human health outcomes. And they estimated that on an average bloom event, a city might save between $55,000 to over $1 million, depending on what type of exposure was avoided. So again, they're using the product not to make their final decisions, but to help them guide their sampling choices and then hopefully reduce exposure. Oregon also uses our product. They created their own Oregon website. So each Monday when the weekly image comes in, they update this website. All their water managers and the public can go and look and see what the water conditions are across Oregon. So they pull that every week. If you're interested in the data and you're not ready to start your own website, that's okay, because we actually have one. It's very intuitive. It's a Google Map interface. You can see I've dropped pins all over. The pins are different colors. Green means the water quality looks good from the satellites. Yellow, orange, red is an indication of how the lakes are doing. Once you drop a pin, you can zoom in. And this is Lake Mendota near Madison. And you can get the most recent satellite image. So it gives you an indication of what's going on across the lake. This is an image from last June. I wanted to get really like near time images, but actually there's not much going on right now, which is good because it's so early in spring. Once you're looking around on the app, if you drop a pin, you can go up to the corner and it says My Locations. If you click on that, it gives you all the recent satellite images. So you can zoom into that whole region and see what's going on. So that's a quick way to get images. Also at the top, you can see there is an option for chart. So this is the last 4 years of data. It goes back to 2020. Each week, you can see what's going on in the lake. And you can see each season there's kind of a summer bloom there. So it's a pretty neat way very quickly to get kind of a summary of what's going on. This is for that single pin drop. So one location in the lake. If you go back to the page and you can see there's a water body stats, then it's going to give you the last 30 days, a summary for the entire lake. Again, this is an image from last June. So you can see at the beginning of May, there's not really any cyanobacteria. If you look at this graph, As we move through May and into June, it goes from a mild bloom to a pretty significant bloom. So you get 30 days summary really quickly, just a couple of clicks, and you can find out all this data on the website. So that's our Cyan website. It's hosted by EPA with the data we make at NASA. If you want a more full summary, EPA actually takes all the Cyan data each year and puts it into EnviroAtlas and in their report on the environment. So you can go to different regions or lakes and get summaries on the temporal frequency of your blooms, the spatial extent, the magnitude, which is kind of a combination of how bad and how long do they last, and then the occurrence. So how many lakes in the region are having blooms? So all of this is done automatically. You can just go to these different sources. They use the Cyan data to make these summary reports. So this is a ton of information because I only had 7 minutes, but we have lots and lots of resources. So you can go to the NASA Cyan page. That's where all the data is. You can download everything. You can explore it yourself. If you go to the EPA page, all the publications, we have publications about every single one of those metrics, anything else that people have been doing with the data. There's lots of training. So that website that I gave a quick tour of, there's a full hour training on that. So you can learn all the bells and whistles. There's a lot you can do. There's free NASA software. You can interact with the data called CDAS. Again, lots of trainings. And then we have something called the Help Hub. So if you want to do like Python tutorials or anything like that to interact with the data, we have a lot of resources available. If you're sitting here thinking 300 meters, that's too big, we have a couple other projects coming. So we're going to start looking at Sentinel-2, which is 20-meter resolution, slightly different bands. So we're trying to figure out what type of product. And then we launched the satellite about a year ago called PACE, which is hyperspectral. So hopefully it will let us know a little bit more about which plankton are where, but that's a 1 kilometer pixel. So that's going to be large lakes only. So that's it. I want to remind everyone, these tiny little things change the world, and we're tiny little things too. So keep that in mind. Get out there and keep fighting. So thank you.
Any quick questions for Dr. Sears before we move on? Sure.
Quick question, Bridget. In Madison, we have this app called Water Lake Forecaster or Lake Tool, and it's a lot of each data available like an SCDF or is there a site you can log and grab that data
Sure.
like on a daily basis and then put it into this package?
Yep, that's a great question. So the data is delivered in 2 different ways. If you want the imagery, like I was showing, we have GeoTIFFs produced daily, and the data also comes in a NetCDF if you want to interact with it that way. And you can also download all of the individual bands that go into the algorithm if you want to look at that data as well. So those are both available. Yep. And every day. So this morning, yesterday's data was put forward.
All right, thanks.
All right, we don't have any questions online, but Bridget, I just had a question for you regarding sorry, I didn't mean to put you down there. Just regarding the resolution for that Sentinel-2 data. So what size lake do you think that would be able to really like cover for the future work?
Yeah, the Sentinel-2 pixels normally are about 20 meters. So
Okay.
into some pretty small lakes.
Smaller lakes, yeah.
Yeah, with the standard product, with Ulchi, with the 300 meter, we can resolve just over 2,000 lakes. And with the Sentinel-2, I think it's over 200,000. So we get into a huge amount of more lakes. The nice thing with Ulchi, even though it's only larger lakes, it's the majority of the drinking water supplies are in those larger lakes. So it's really helpful with that and a lot of the recreational, but there's needs for the small lakes too, especially the narrow reservoirs that can be challenging with the standard product as well.
Perfect. Thank you, Bridget. All right, so the other question we had, it was so just more of a statement. It's a little difficult to hear the in-room questions, so if we can make sure that the mic is held up to the person asking the question, that'll be really helpful. Okay, I'd like to introduce our next speaker, Dr. Regan Herrera from NOAA. Dr. Herrera's research focuses on phytoplankton ecology, concentrating on harmful algal species and global climate change. Her interests in HABs include identifying factors influencing toxin production, allelopathic chemicals, and HABs' impact on the phytoplankton community dynamics. Dr. Herrera's research also explores how the impact of acidification, temperature, and timing of freshwater inflows impacts the phytoplankton community. In addition, Dr. Herrera has also had the opportunity to work worldwide, including coastal Louisiana and Texas, the Western Indian Ocean, Texas reservoirs, Brenton Sound estuary. I hope I pronounce this right. Barataria Lake.
Barataria.
Thank you. And the Great Lakes. So please welcome Dr. Herrera.
Everyone can see my slides okay? Slide check.
Yep.
Okay, excellent. Thanks, and I'm sorry that I'm coming to you virtually. I would have loved to be there, but that was out of my hands. But thanks for still having us and accommodating the change. So I work at the NOAA Great Lakes Environmental Research Laboratory, which is in Ann Arbor, Michigan, where we do lots of different type of research, including anything from ice to harmful algal bloom ecology and research. So what I'm going to talk to you a little bit about today is some of our work that we do monitoring blooms within the Great Lakes itself. So could I also get a check that it advanced? Okay. Maybe.
Yep. Yeah, that worked.
Okay. Excellent. Thank you. So our overall forecasting and detection systems range a whole bunch of different products. So we do everything from satellite imaging, kind of some of the stuff that Bridget talked about just recently, as well as do all the way to the end, which is what I consider the experiments that we do, both field and lab-based experiments to look at HABs across the region. I'm only going to talk about a few of these today, which is our hyperspectral camera and then our environmental sample processors and AUVs. All of the data that I'm going to talk about is either available through our Lake Erie forecast, which is hosted by NCOS, which is a part of NOAA, or through our water quality data, which is housed here at GLERL, which is also accessible from the website. Some of this data too is also available through the Great Lakes Observing System, or GLOSS, which is a regional portion of IUS. So when we look at the Great Lakes as a whole, anyone who lives in the Great Lakes will know that we have a lot of cloudy days. In fact, I think it's estimated that 205 days per year in the state of Michigan are cloudy. And that really then hinders the availability of getting good satellite imaging on areas in which HABs are occurring. And one way around this, in which we have started to kind of tackle this issue and still be able to produce good quality reports, especially for our water managers and our beach managers, is through hyperspectral imaging. And we do this through two ways, mainly through crewed platform flights, in which we send up a hyperspectral camera. You can kind of see up in the upper corner. This sits on that crewed flight. They go and do many different types of flights. They can be just transects, which is kind of in the middle, which I'm showing, or what we do is mow the lawn, which is the one over to the right-hand side pictured, in which we go back and forth and kind of make a full-on picture of what the bloom is looking like. Once we get that back into the lab, those images, we actually have an algorithm that can pull out the cyanobacteria from there and produce reports to see how thick or how dense the cyanobacteria is within that area. So we do this weekly for the water managers, both in Ohio and Michigan, both in Lake Erie and Saginaw Bay, and this helps determine where the bloom is located, especially on those cloudy days. We can also do this to help us guide where we're going to go sample. So a lot of times when we're out sampling through our ship crewed monitoring programs, we will use these flights to determine where to go and sample within the system to then determine the toxicity of the bloom. And then I'll show another example here in a minute of how we use this. Oh, I forgot that this was going to pop up in that with these mow the lawn type scenarios, we can then find those hot spots and when we get to the hot spot, lo and behold, we have nice scum, typically, which we can then sample and determine toxin. We also use the hyperspectral camera on what we call uncrewed platforms. This is what typically we know as a drone. These again are attached to these uncrewed platforms, and then we can send them out into the field. So I'm just showing here from the satellite imaging what we get from Ulchi, which again Bridget just nicely explained, versus our crewed flights, which is about a 1 meter resolution, up to our then drone flights, which is about a 10 centimeter resolution. It really gets at the dynamics of the bloom itself, of how we can find really key areas where scums might be forming, which are going to then increase the potential for exposure to toxin. We also have uncrewed systems that we put into the lake, which are our environmental sample processors. And we have a network of these. So we have 3 second-generation environmental sample processors here at the laboratory. These are also known as labs in the can. They basically do an ELISA in situ in the lake for us. So we have trained the immunoassay to recognize microcystin toxins or particulate microcystin toxins, and it does it within 2 hours of water of collection. So what we call this is near real-time sampling. It can also collect omics parameters, which I'll get a little bit more here into in a second. But these landers, so the actual instrument, which is over to the right, you can see the innards of it. It does lots of microfluidic exchanges, which I can get into if you have any questions. We then put this in a lander or a can, which you can kind of see in the little picture and the little drawing. And this sits at the bottom of the lake. And this allows us then to take 2 different types of samples, one at the bottom of the lake and then one at the surface. We can also do intermediate if we so choose. But right now, those are our 2 focuses. We have 2 locations that this sits. One is what we call the Ohio coast, or right near the Toledo water intake. And the other one is on the Michigan coast near Monroe, Michigan. And what you can see from this data is now we can get basically 24-hour data if we choose, or what we typically do is take a reading once per day at these 2 different locations. It really helps fill in the holes of how quickly the toxin production can change within the lake itself. In fact, we can capture as high as a 650-fold increase in toxin over a 48-hour period. And then that allows us to then send out warnings that the toxins are increasing within a certain area. However, if you've ever been in a cyanobacteria bloom, you know it looks like this. So stationary locations are great, but really we have toxin changing spatially as well. So we've worked with Embari to help shrink down that 2G ESP into a smaller device. So now we again get near real-time toxin analysis through an SBR module, and then also omics type parameters, mainly genetics that we're looking at through an archive process. And we can then put this device, this 3G or 3rd-generation ESP, into a long-range AUV. And you can kind of see them 2 next to each other, the 2G versus the 3G. We also typically have sent this out with a Sentinel device, which then goes and finds hotspots of chlorophyll A and it sends the 3G ESP to that location and takes a sample. We've tested this system both in 2018, 19, and 2021, all very successful determinations and tests of this type of technology. However, we also know it's very limited. So a long-range AUV is great in deep systems. Most of the systems in which our blooms occur are not deep. And the human interaction with the HAB is greatest in these shallow water areas. And what I'm showing on the left is that long-range AUV that we actually got stuck in the mud because we tried to go too shallow and had to go get it out. So wouldn't it be great if we could get into those shallow locations and provide toxin reports there? And we are. So we've developed what is known as the SHARC, which is the Surface Harmful Algal Bloom Research Craft, which takes that 3G ESP and actually puts it in the hull of this surface AUV or autonomous vehicle. and goes out and samples. So we can direct this autonomous vehicle in Ann Arbor when it's on Lake Erie through computers. We can tow it behind a boat, or we can actually use a remote control, which is kind of, kind of thinking when you're back in a kid wanting to remote control a boat around the lakes. We can actually do that with this as well. And we've sent this out in 2003 with a very successful deployment, and was able to get very shallow areas with testing with the 3G ESP. In 2024, we went out and sampled again. This time, we actually combined the drone technology with this autonomous surface vehicle technology, and the drone was able to take images of where hotspots were, and then we were able to send the autonomous or the shark to those locations to do that sampling. Unfortunately, we had a collision with a private vessel in 2024 that has put the shark currently out of commission. So we were unable to finish that um, deployment overall. Um, at this point, I kind of just left it open to questions if you guys have anything. If not, I can wait for the panel. I'm going to exit out of my presentation if that's cool.
Sure. Any questions for Dr. Herrera? Not right now. Do you have any online, Pam?
None online, but I do because
Okay.
these are super cool, innovative instruments to look at these, the differences in HAB, especially spatially and temporally, right? So
Mhm.
I was wondering for the 2G ESP, how many, does it sample by using by sample cartridge, and how often does it need to be changed? And how many cartridges does it hold?
Yep, that's a great question. So it does have cartridges. It takes 3 cartridges, or what we would call actually pucks,
Okay.
per sample. And then typically we can use that for 44 samples total. And that includes a positive and a negative both before we can and after we can, or after it comes back to make sure that we are getting correct toxin levels and we don't have to flag anything. Um, that can be spread out. So typically we leave ours out for about 20 to 22 days. And that is because we're sampling both at the surface and the bottom of the lake every day. Well, the bottom every other day, the surface every day. So it just, you can also change that frequency of sampling on the fly. And I should say that that's true for both systems. So both the 2G and the 3G. If you decide that you want to sample someplace different, you can go ahead and send that information to the instrument and it'll sample as needed.
And just one more question. Then does it report data, or do you have to download it? So is it telemetered, or do you have to download the data?
So for the 2G ESP, you actually download an image, and from that image, we analyze the toxin. So each time it runs a sample, the reason it takes 3 pucks is because we also have to do a control. So each sample also has a control related with it, and from there we analyze that and send it out. So that's where that near real time, because it takes about an hour and a half to run all the processes. We get the image, we analyze it, and then put the data out. For the 3G ESP, it takes about an hour, and that again, it pings us and sends us not an image this time, but a graph, and from that we can analyze based off of the standards, and then put that number out.
Thank you very much.
Yep.
All right, great. So our next speaker is Matt Titus. Matt is a data scientist at Clearwater Analytica, a public benefit company focused on real-time monitoring and forecasting of harmful algal blooms in sources of drinking water. Dr. Titus develops remote sensing data products and AI tools, forecasting growth and decay of algal populations using a combination of public and private data, both satellite and in situ. He has served as a co-PI on a Water Resource Foundation grant, applying explainable artificial intelligence to study algal bloom risk in a variety of watersheds across the US and Canada. So thank you very much. Oh. One moment, just learning to share our screen again.
Just so people can see. Thank you. Yeah, thanks to the organizers for having me. I'm Matthew Titus of Clearwater Analytica. I'm a data science consultant, and so I suffer from a degree in mathematics, so there's a little bit of a language barrier sometimes. But I've been at this for a few years. I got into HAB science through an emergency event that happened close to where I live. I spent a few years living in Salem, and then while I was in Corvallis doing a postdoc at OSU, there was a harmful algal bloom at Detroit Lake that affected the water supply of several municipalities. And so they reached out. They were looking for short-term solutions to getting an early warning signal for harmful algal blooms. So they don't, the Army Corps manages that lake, so there's very little interventions that the municipalities could do. And so they just wanted to have a forecast so that they could see it coming down the line and begin to prepare at their plant and inform the public. So we approached this with and I apologize, I'm working with a PDF, so some of this is getting covered, but we used a Bayesian model averaging technique to try and get this forecast out. Essentially, that allows you to use as many modes of data as you have available. And the idea is essentially that even though there isn't a single answer, there's no one data set that's going to really key you in on what's going to create the bloom in the next 7 to 14 days. Each of these models might have some lens in on the problem. And so creating kind of a wisdom of crowds approach is what the BMA is after. The nice thing about that is if one data set drops out, if it takes a little bit longer to get your nutrients analyzed or what have you, the models that rely on that data will not be able to operate. But all of the other models, things that might depend on weather or remote sensing data, continue to give you a prediction. So there's a resilience to that approach. One question that we had, so at Detroit Lake, for example, we got about 85% accuracy in predicting next week's bloom state. You can imagine a lot of that's an autocorrelative effect, but we saw really distinct impacts from nutrients. So phosphorus was a huge driver for Detroit Lake at that time. A couple of things have happened around the lake that might change that. But we wanted to do this nationwide. We wanted to see if there was a modeling approach where we could do something similar, capture the dynamics of the bloom in environmental variables so that we could tell people, this is what you should be looking for in your watershed. So we worked with the Water Research Foundation. We applied for a grant and received it back in 2021, and we were working on explainable artificial intelligence. So the idea is simply that, I'm not at the stage of trust with AI that I'm going to climb into my car and say, take me to Houston and just go to sleep in the driver's seat. So you want to be able to work your way back and figure out why that model is telling you what it's telling you. And so that's what this project was about. Wanting to build trust in people that were stakeholders or utility managers in what the model results were. And so the basis for this project, we were using decision trees and building random forest models. And you can see kind of a schematic of what that looks like in that upper left figure. The splitting between each of these sets is looking at a variable and is thresholding it. If you're above this level, go left. If you're below it, go right. And you move through the tree in that fashion. When you get to the bottom, you'll see a predicted value. What's on the right is this pinwheel it's a collection of maybe 50 some of these models. And the ones that are highlighted are those whose conditions are currently being met with by the variable values at that time. This is nice because you can plug in other variable values and get a modified prediction that lets you run counterfactuals. The other nice thing about it is you can summarize the influence of each variable. So if I were to increase the chlorophyta abundance by some amount, what effect would that have on next week's cyanobacteria abundance? You can run through that. So that's summarized in partial dependence plots. I won't be able to do a demo now, but if you're interested, please get in touch. So this was fun, but it did teach us that there is a really big it's a very idiomatic problem. So there were not watershed models that were transportable between separate watersheds, which is an indicator that all of our modeling has a ways to go in terms of pulling out features that are going to be consistent between New York and Texas and Oregon. These places all function on the same basic biophysical principles, but the data reflects those dynamics in very different ways. And then moving to remote sensing data and modeling based on that, we wanted to have a national product, something that would tell us in every resolvable lake what the probability of a bloom would be. And so neural network model was trained, and it has pretty decent accuracy, pretty decent R squared. It's now been superseded. The EPA has come for us. The published paper last year with Blake Schaefer, who is one of the other big brains behind the cyan work. So what this model does is it takes in that cyan cell count estimate, and it also takes in physical attributes of the lake and then the local weather data. And using that tries to predict what that cyan count estimate will be next week. So it's a cyan of the future. And so Blake and friends did this last year as well and did a really good job of it. So this is also something that will likely become publicly available if it's not already. But yeah, Clearwater, we did this, I think, in 2021. And one of the difficulties that we found was, I mean, I love cyan and it's an amazing tool. But there is a it's been mentioned once or twice, the resolution of the cyan data product, I think it comes from Sentinel-3, is a 300-meter resolution. So each pixel is 300 meters by 300 meters. A lot of mid-sized lakes won't be resolved by that. One of the issues is you get stray light from the shoreline. And so if you have a pixel that's next to the shore, you can get false positives, you can get false readings of cyanobacteria being there. And so what the standard operating procedure is to kind of cull those shoreline pixels, which means that if you want to get data, you need to have a 900 meter by 900 meter space in the center of your lake. And a lot of lakes just don't have that. So there's this work that's been done. I was put in touch with Abhishek Kumar, who I heard about his work through Amalia Handler, who I believe is another EPA voice. And so big thanks to both of them for the connections and the work. They've done this research, which is really, I've been really impressed and delighted by it. I don't know if you can make it out on this figure, but there's this brown curve going across all of these Roman numerals. The x-axis here represents wavelengths, and the y-axis is kind of a sensitivity to those wavelengths. And so different bands are resolved on the instruments on, this is for Landsat 8, the OLI instrument. And so, for example, band 2 is going to be yellow-green wavelengths, band 4 is reds. If you subtract those from what's called the panchromatic band, which is the long brown one that I pointed out, you're left with region 1 and 3, which is kind of a blue-green light, some pollution from that. And then the orange light that is at that Roman numeral 3. So even though Sentinel-8 doesn't have a sensor for this, you can still get the data out, and it lets you infer cyanobacteria's absorption of light at that frequency, which is what phycocyanin does. That specific pigment is absorbing in that region. So this is a way to kind of get a backdoor into what the Cyan project does. And so you get this upscale. 300 meters to 30 meters is maybe hard to visualize. It's happening in the X direction and the Y. So it's a 100-fold increase in the number of pixels you're resolving. So this left photo, Clearwater is a big consumer of cyan data. So the left photos are hosting of cyan data. And then the right photos, a depiction of this virtual orange band that's used for detecting phycocyanin. So this is nice for those mid-sized lakes that are trying to address the same problem, but just can't quite get that visualization. And I should also say that this problem will be, I'm sure, tackled by the EPA and NOAA and NASA in the early 2030s. Landsat Next is another mission that's going to go up, and it's going to have both the hyperspectral bands that are relevant for phycocyanin, and it'll have it at higher resolution. So this is just getting better and better on the data side. And then I should quit talking, but I just want to throw out there that, yeah, there's a lot of future research on the analysis side that I'm very interested in doing. There is a lot of feature engineering that I think we're missing, and that's evidenced by the fact that we didn't find transportable models when we looked from watershed to watershed. But I want to thank everybody for their time.
Awesome. Thank you. Any live audience questions for Matt? I have one. I was wondering with the grant that you went over first from the Water Research Foundation. So once you've made that product, are the users for that, like who are they, and are they continuing to use it, like taking their data to predictions, or?
Yeah, the project was done through the WRF. So if you're a WRF member, you can go to the project page. It's project number 5080. And you can look at the web tool that was created. The tool is underutilized in the sense that you could set it up to accept a CSV of data and have it adjust that wheel and say, these are the variable values. Now let me get my prediction of cells per mil. I don't have the ability to do that quickly and easily. And so it wasn't done as part of the project. The project was largely research and exploratory. But there is that potential there. If you wanted to see what your data would suggest is going to happen next, there are 5 lakes on that project, the web tool there. And so you could find one and you say, oh, this is kind of like Alpine region that I'm interested in, and see if you can manipulate those variables. But I think that there's a lot of meat left on the bone there in terms of building a user interface where someone could use that and have it online all the time.
All right, thanks. Pam, do you have any online questions?
Yes, we have one from Nicole Manasco from the Army Corps, I believe, or USACE. How is explainable AI different or similar to supervised AI?
Great question. So supervised AI is a general term for when we train a model where a human's gone through and collected a validated data set, and we're using the human's training data to tell the model what to do and then validate against it. It's very typical, but you see this done with neural networks. The neural network for cyan data is essentially a supervised learning model where we put in all the data from week 1, and then at the following week, we gather its cyan readings, and we use that as the answer. And so it's being validated against that. It's being supervised and being told what the correct answer is. It's not just clustering or something like that. So that process is, you can see that in all sorts of black box systems like a neural network. The explainable AI specifically is giving you the ability to quantify how much a variable is affecting your model. And you can even do it for specific conditions. So for example, the impact of additional nitrogen in your system might vary depending on what state your system is in. Some states you might add more nitrogen and have no impact on the algae, and other times you might see a huge influx. And so being able to pull out what the impact of individual variables is on the prediction itself is what's different there. I hope that was super wordy, but I hope that got across the spirit of the answer.
I understood.
Thank you.
Thanks. Okay, next is Tyler. We're gonna go set up. So while Tyler's setting up, Tyler graduated from the Ohio State University in 2014, where he worked in the aquatic ecology lab and focused on water quality sensors and control systems. He has since worked in aquaculture facilities and served as a field service engineer at YSI within their remote monitoring systems group. At YSI, he worked with water quality buoys, vertical profilers, stream gauges, and many more sensor packages. Tyler currently works as a watershed supervisor for Veolia, a private water company in the American Northeast, serving 700,000 clients. Veolia is actively developing scalable models to predict, predict, prevent, detect, and treat harmful algal blooms and other water quality concerns using in situ monitoring instruments, satellite imagery, and AI. So 1 second, and we should be ready to go.
Hey, good morning, everyone. We got to see how they made all this sweet technology today, and now you get to see how we put it to work for our utility. First thing we had to do was justify why we needed to put all these sensors in and acquire all of this data from satellites and all of these other public data sources. For us, it's a matter of sustainability. Our operations depend on adapting to a changing climate, and remote monitoring gives us the necessary tools to do that. Right now, the data informs models that Dr. Titus and a number of other people have developed for us. Those give us the ability to extract meaningful insights from data sets that are honestly more complex than I know what to do with. They also make it understandable for our public stakeholders within our communities so they can support our management decisions, because there is frequently a lot of headbutting in town halls over some of the things we'd like to do. They also are able to they enable us to adjust our management strategies in something closer to real time, which tends to save money on operations and allow us to allocate our labor more effectively. I guess there's the benefit of greater sample collection, the resolution, and improved accuracy. The removal of human error is a benefit to us, but we do still have to validate all of our remote sensing equipment with field samples. So to give you an idea what we're working with, we have 3 lakes in our watershed that we're responsible for managing. The largest of the 3 is the Oradell Reservoir. That's where our water treatment plant is. That's kind of where all of the remote network started. So first thing we got was a vertical profiler, so we can determine degrees of stratification within the lake. We have sensors on our intake and in our DAF process, so we can determine how efficiently we're removing things from the water. Our lab samples 2 to 3 times a week throughout the growing season, and every 2 weeks when it's not. And then we have all of the process controls within our plant, so we can evaluate the performance of our plant and try to figure out what the trend between source water quality and plant performance is. And recently we added satellite imagery to that. On the other lakes, we have just standard surface water quality buoys measuring all of your standard parameters, lab sampling, and satellite imagery as well. So this is Oradell, to give you an idea how we've got it distributed. We tried to target places where we have sampling points so we can actually validate the data we're getting. And we try to include something to measure near any of our tributaries and diversions so we can see what to expect coming in from upstream. Big thanks to USGS for your stream gauges, because we would have had to install a lot of those if they weren't already there. So the ways we use this data, one of our first projects was nutrient mitigation. So we knew, I mean, from anecdotal evidence, that we had a lot of sources of nutrient pollution coming in from around the watershed. The issue was we didn't know where they were, so we didn't know where to deploy our mitigation strategies. But Dr. Titus and Dr. Seegers, with their technology, we ended up with our own Clearwater dashboard. And through this, we could see our chlorophyll hotspots and have a better idea of where we actually needed to deploy these things. So last year, we applied something called Utrizorb G to one of our smaller lakes. It's Woodcliff Lake. It's been pretty heavily impacted as long as I've been able to find reports on it. And it's usually where we see the worst HABs start to happen in our watershed. The Utrizorb is designed to drop through the water column, land in the sediment, and sequester phosphorus. And based on the results we've seen from the last growing season, that mitigation was effective. And through continuous monitoring with the satellite imagery, we can determine when we need to add more Utrizorb, or if there are other mitigation strategies we can deploy that would prevent sediment nutrient levels from increasing again. We're looking into biochar now. That seems to be one that's becoming more popular for stripping the water column rather than the sediment of nutrient pollution. And those deployments are going to be designed based on where we see the hotspots as well. I mentioned the vertical profiler. That was actually started because we needed to figure out how to get a better aeration system in our lake. And to do that, we needed to know how bad the stratification was. I'm sure you're all familiar with what a vertical profiler is. You probably talked to the Xyleman Institute guys already. But this gave us the ability to determine what type of aeration system would be most effective. Back in the 80s, I think, they installed the big like floating tower aeration systems, which broke constantly and didn't really disperse dissolved oxygen as well as we needed it to in the lake. So with the data we collected from the vertical profiler, we were able to design a much better grid system that goes across the bottom of the lake, 53 different diffusers, and we've seen significantly fewer incidences of hypoxia at lake bottom since doing this. This data also goes into Matt's Clearwater model. So to give you an idea what all goes into that, we use the Cyan project, we use our lab counts, the surface buoys, the vertical profiler. We're looking to add the plant performance data. I think the philosophy we're trying to take with it is viewing the plant as an extension of the water body rather than the water body as an extension of the plant. And then we use open source meteorological data and the stream gauges. And through all of this, we get two different models that can report different things about what to expect about an upcoming HAB. The first is the Cyan model, which is like a risk forecast. It's the likelihood of seeing a HAB. And then we have the BMA model, which evaluates what density we should expect the HAB to be. So more of an intensity rather than a yes or no. And it also evaluates historical satellite imagery to determine when we could expect it based on a temporal component rather than a physiochemical one. So as far as how we use these HABs forecasts, for us, it's a matter of labor. As you can see from this particular example, July 4th was a perfect time for an algae treatment, and nobody on my team wants to be out on the lake doing an algae treatment on July 4th. So since we had time and saw it coming, we were able to plan around that a little better and effectively reduce taste and odor compounds and eliminate a pretty large portion of the cyanobacteria in the water. I can't read this at all. I should have made this bigger. I'm sorry. In general, up top, you can see the satellite images for cyan though. So we saw the blooms start to form at some of our tributaries. The treatment happened sometime around July 5th. It attenuated for about 10 days. And as it frequently does in New Jersey, within about 2 weeks, we had another bloom coming. Again, had to plan for that. The great thing about the spatial component is that we can target our treatments. By regulations, we still have to treat half of the surface acreage of the lake, but we can choose which half of the lake we're treating. So if we know the HABs are most prevalent in the northern part of the lake where the lagoons are, we can treat up there. Typically what we find is they're more near the center of the lake body, which conveniently is where our plant intake is as well. So just to dig a little deeper into the instrumentation technology we're using, this is what a vertical profiler looks like. The BGA sensor is one of my personal favorites because we can see the actual like diurnal migration of these phytoplankton moving up and down through the water column with the sun. We've considered maybe deploying like a targeted depth level treatment rather than just surface treatments. I think there might be some regulations that prevent us from doing that at the moment, but it's worth exploring the option, I guess. Surface buoys, I would just have dozens of these if I could. The lake would be covered in them, but they give us like really useful local data so we can determine what's happening. If we get a report of a fish kill from the field, there's usually a buoy nearby that we can check to see if we can start to investigate the cause of it. The plant sensors are, necessary to make sure your plant's working, but they also reduce downtime because we can determine, which instruments are working properly, what parts of the process might be suffering. And frequently we can tie it to source water quality. So if we see our filter run times decline, we usually know that it's because we have algae coming in, and we can confirm that with our numerous other sources from the field. And lastly, the big project for this year is going to be trying to pair the lab counts with the satellite imagery. We're just trying to lock that correlation in as well as we can, so the estimates we get on our Clearwater dashboard are as close to real life as they can be. And that is all I have for you. Do you guys have any questions?
All right, thank you, Tyler. Anybody in the room have questions? No? All right, I got one. Just wondering, like, so since you have the dashboard, when you come in the morning, on a typical work day, I assume you check the dashboard, and then how much of like the raw data from your continuous monitors, like, are you checking? Do you just look at the dashboard and make decisions off of that, or?
So we look at the raw data pretty consistently as well. The dashboard is a great tool for, I guess, the higher level decision makers within the company. They don't necessarily know what a lot of these things mean. So for us, it's packaging it in a way that's easily digestible, whether it's through public officials with municipalities that are within our community, or if it's, senior level executives at the company that are determining whether or not to fund a project we've identified that might be helpful.
All right, great. Pam, do you have anything? Oh, we got one more in person. Cool, cool, cool.
How are you using the FDOM data?
To be honest with you, I haven't dug into that parameter as much as I'd like to. If anyone has suggestions on what FDOM might be telling me, I'm all ears.
Pam, do you have anything online?
That was also a question from online, is how useful have you found your FDOM meter, your data?
It's generally the messier data. I think
Yeah.
the doubt we have for that data is probably the reason we haven't tried to make more use of it. I think the calibration tends to be challenging for a lot of people, including our instrumentation team. So when I'm seeing consistent negative FDOM numbers, it kind of just seems like a throwaway to me.
Nope. We got one more question online from Chris Lee. What about a DO sensor?
Oh, the DO sensor is the best, absolute favorite. That's how we know when the hypoxia is happening. It's how we know when to expect, all the decomposers at the bottom of the lake to start accelerating. It's always an exponential thing. It starts as something small. You'll see it drop from, 7 to 6.5. And within 2 days, it could be down to 2.5. Unfortunately, with the new aeration system, we haven't had that happen in a while.
Okay, great.
All right, so I'm going to introduce our next speaker, Dr. Lauren Weinrich she is the Director of Research and Development at American Water based in Camden, New Jersey. Lauren has a passion for improving water quality, investigating contaminants of concern, and engaging internal and external stakeholders to address current and future issues facing the water industry. She is a contributing member of various professional technical advisory work groups through the American Waterworks Association, including the Real-Time Water Quality Monitoring Committee, and serves as deputy editor for AWWA's journal Water Science. She holds a bachelor's in biology from Marymount University, a master's in environmental science and engineering from UNC Chapel Hill, and a doctorate in environmental science and engineering from Drexel University. So please welcome Dr. Weinrich.
Great. Thanks so much, Pam. Great to see everyone. It's been a really exciting session so far. There's lots to dig into. Just want to start my timer so I don't go too long here. The first part that I want to focus on, so being at American Water, just to kind of level set, hearing from Tyler and Veolia and the water drinking water aspect of things, kind of honing into how we think about, our systems and supplies. As an overview, if you're not familiar, American Water is the largest water and wastewater company in the U.S. And so I'm showing this map here because you can see, based on the footprint, the the sources of water supply are varied. I will kind of hone in this talk a little bit on the Iowa and Illinois area in the Midwest. That is around the Mississippi River. So different sources of supply for our 80 surface water treatment plants include rivers, reservoirs, and lakes. So there are numerous challenges, of course, that come with that. But, given the technologies that are out there and treatment for managing cyanotoxin and HABs, we've started a new project that I'll talk about at the end as well. Kind of first starting to think about, though, the source water protection aspect, because of the various sources of supply as well as the risks that are associated. We have just revamped our source water protection group at American Water that I wanted to highlight. We think about this as a multi-barrier approach. So whether it's HABs or other chemical risks, there's a extensive amount of monitoring that needs to be done. Our surface water systems, like I said, there's about 80 or so across our footprint, have assessed these risks based on their location, the chemical risk that could be upstream, the risk for algal blooms, et cetera, and have developed monitoring plans based on that. Many of them include online monitoring. In fact, we were just talking about the FDOM, and other sensors that, depending on the risk, we need to deploy those sensors that can help, give some warning to those risks. The challenge there is the specificity for that monitoring tool that is deployed, as well as the monitoring and maintenance and cleaning of those sensors. Of course, in surface water, whether we have kind of the in situ sonde or even a trough setup for source water incoming, there's a lot of challenges with that. And so quality of data is something that we're extremely focused on and looking at, at all of our systems and improving best practices in order to do that. So just kind of high level, and also thinking about the other tools that we use, including WaterSuite, as considering our zones of concern. Again, this is somewhat more around chemical risks to our supplies, but it really becomes a partnership. And I think what we've, seen today, and I'm excited to be a part of this, is to continue the partnerships that we have between these different agencies and, partnering on research to protect our watersheds and make those improvements that are needed. Again, whether, the program includes risk assessments, characterization, different geographic footprints present different geographic risks, of course. But today we are here to talk about harmful algal blooms and monitoring. So I do want to highlight one event that happened last year in many of the reservoirs where we've seen routine algal blooms, thinking, in our footprint in Pennsylvania and New Jersey, we have a lot of source water control programs in terms of treatment and monitoring that have been put in place. Those are considered high risk, but we had somewhat of a surprise last year when at some of our routine monitoring locations in what we classified as a typically or historically low risk source of supply, we actually were getting cyanotoxin hits on some routine monitoring that was being done at our Illinois intakes. So Illinois American Water has 3 different water treatment plants that are served by the Mississippi River. And then we also have 1 water treatment plant in Iowa that is as well. So given the low risk, the monitoring plan there is generally weekly using strip tests. And I'll show that a little bit more in the next slide. Because there were inconsistencies in the readouts, we basically stood up a crisis response team, assuming worst case scenario that we were on the leading edge of what could have been potentially a major cyanotoxin bloom forming event. That preparation did not go to waste. We had the emergency response team, multi-state and corporate level involvement for chemists at our central lab, where we were able to get sample turnaround in a day. We needed that data for fast decision making. The online monitoring at that point wasn't giving anything necessarily out of the ordinary. I am showing the closest USGS gauge, and it showed some increases of chlorophyll A, but no other visible bloom was happening, nor through, visualization or online data. So regardless, we got staffed up, set up, and prepared with additional treatment, and also messaging plans in place if it became a bloom. So showing here, we mentioned the Water Research Foundation, either I think Matthew did. The image shown on the right is a recent project 5120 that was done. I believe that this is publicly available through the Public Plus option, where Hazen and Sawyer led the project and showed these individual kind of field guides for guidance from test strip kits to online monitoring and more advanced analytical laboratory support. So showing here with the test strip kits, what we found was that we were actually getting hits and then non-hits on daily and multiple samples during the day for cyanotox or for cylindrospermopsin and microcystin, but it wasn't consistent. When we then started the sampling campaign, sent those samples to the lab. In general, we still thought that perhaps this was the start of a bloom. Conditions were favorable, it was warm, lack of rain. Last year it was dry. And so, we were standing up resources in order to respond to this. What we ended up finding was, in fact, that, the ability to be prepared with other data, which generally at this point came through the laboratory. We were running ELISA and methods 545 and 546 through the labs, made treatment adjustments, had to quickly work with supply chain to get additional powdered activated carbon to absorb cyanotoxins. As it turned out, the cyanotoxin levels never reached more than maybe about 2 micrograms per liter for microcystins. We did not see any cylindrospermopsin despite the test strips showing it. So that laboratory validation was extremely important. The levels changed slightly in the Mississippi, increasing, like I said, to maybe about 2. We were able to control any of that in the treatment plant being continually below detection in the treated effluent. So in the grand scheme of things, it was relatively a non-event, but again, it was a very eye-opening event in which all of the routine monitoring done and a historically low risk source of supply, the Mississippi River, we didn't expect it. So a lot that came out of that, a lot of lessons learned, and in fact, a revision to our cyanotoxin management plan with updated assessments. The other part that I wanted to mention today is a project we just started last year with the Water Research Foundation. This is project 5226. We are looking at various aspects of from satellite monitoring down to genetic sequencing and all steps in between. Definitely would love to connect with anyone else interested in this research or have done similarly. We recently acquired a hyperspectral microscope ourselves to start conducting additional insights into those species that are HAB forming and those cyanobacteria. So part of this is just the overview. I believe it's online as well, showing the different tiered analysis that we'll be including. We have multiple surface water sources involved in this project, including ours at American Water and other partners across the United States, including Texas and some other high bloom activity areas. So a lot more to come. And again, looking forward to partnership on this and any other engagement along the way. And I believe that is last slide. So want to thank everyone.
Okay, any in the room questions for Dr. Weinrich? Yep.
I was curious about the limited detection on the test strip and then the in laboratory, I think you said Eliza, for looking at the cylinders for mopsin.
So thanks for the question. Yes, the test strip limit, I believe, is 0.5. So that presents a challenge because the drinking water limit for higher risk is 0.3. The laboratory-based detection limit is lower than that. We could look at instrument values, but again, in terms of information, the biggest challenge with the test strips was that detection limit. So in recreational spaces, it makes a lot more sense. For drinking water monitoring, there are some challenges with it. And in fact, in that project 5120, some of the pros and cons that the field guide shows relate to detection limits, the application, considering those limits and criteria that are needed.
Great. Anybody else? Nope. Got anybody online, Pam?
There's a long question. I'm going to think about this, and we might bring it up after Jessica's talk.
Okay.
Okay. So I'll go ahead. Thank you very much, Lauren. So I'm going to take this opportunity to introduce Jessica Sakali. Jessica works for the USGS. She received her Bachelor in Science degree in microbiology from Ohio State University also, and has been working for the USGS since 2014 at the Ohio Water Science Center. She is the Ohio Water Microbiology Laboratory coordinator and works on multiple integrated water quality research projects. Jessica has worked on studies identifying factors related to toxic cyanobacteria blooms, developing a HAB early warning nowcast system for Lake Erie, identification of microcystin degrading bacteria, and is currently working on integrating cyan satellite products into predictive models for cyanotoxins. So let's please welcome Jessica.
All right, thank you. Can you see my slides?
Not yet.
All right, this one.
Yep. Now we see him.
All right, thank you. Okay, so thank you again for having me here. I'm excited to talk about predictive modeling for HABs at Ohio inland lakes and the integration of cyanobacteria assessment network satellite estimates into the models. And I'm also doing this work along with Aaron Stelzer and Lauren Lynch in the Ohio Water Microbiology Lab, and then Keith Lofton is another USGS Cyan lead as well. So as you may all already know, cyanohabs major water quality concern, and in Lake Erie in 2014, Ohio had a major bloom, and the city of Toledo had to close down the water treatment plant. People didn't have water for 3 days, and it affected about 400,000 people. And while Ohio hasn't had major problems since then, affecting as many people, we're still getting toxic blooms every summer in the inland lakes and Lake Erie as well. So there's still a need for the early warning systems. And Dr. Seegers kind of already introduced Cyanobacteria Assessment Network, but this is a collaborative project with USGS, US EPA, NOAA, and NASA to identify those cyanobacteria blooms in lakes and develop an early warning system. So this project specifically for USGS, the samples we collect are helping to ground truth the satellite algorithms and refine any of the algorithms as well. And we're also developing predictive models because while the satellite data can tell you if the blooms are occurring, they're not telling you specifically if they're toxic or actively producing toxins. So that's what USGS is developing these additional predictive models for. And then all of the sample data and everything from this project is also going into the Enviro Atlas web and mobile app. And then it's going to be available publicly on USGS NWIS, which is the National Water Information System. And then just a little bit more of the project background. In Ohio Water Microbiology Lab, we've been modeling for a long time. First was E. coli in 1998. And then we started using virtual beach in about 2011 due to the statistical models outperforming persistence methods. And we also applied that to try to predict cyanotoxin threshold exceedance for predicting models so that people will know whether the water is toxic or not. And then in 2013 through 2019, we've been working on Lake Erie and then specific inland lakes in Ohio. And then this project is expanding to new lakes that we haven't modeled at before. So these lakes are Cedar Creek Lake and Buck Creek Lake. And we're sampling these during the satellite flyovers. And we're taking water quality measurements at 4 sites across the lake. And we have a sonde, so we're doing dissolved oxygen, turbidity, specific conductance, all those. And then also laboratory analyses, cyanobacteria genes by qPCR, the toxin concentrations obtained by ELISA, and then nutrient concentrations as well. The satellite platform used was the Olchi Sentinel 3A and 3B. And the data collection was from 2020 to 2021, and then from 2024 and then 2025 as well. And there are some limitations for the satellite data already covered in previous presentations, but again, we do have to take into account the land versus water, mixed pixels, and these lakes are resolvable down to 300 meters. So they're able to be resolved by these satellites. For the multiple linear regression models, we use Virtual Beach. It's a free software developed by EPA. These build the site-specific models that have 2 or more variables and then also multiple years of data. So we start off with at least 2 years of data and then validate the models with at least 4 years of data. And these are used, again, to predict a threshold of exceedance of the toxin. So we've built models for all 4 toxins if those are present in the lake or whatever you're testing. But most of the time it ends up being a microcystin. And then these simple models can be used to make management decisions so that water managers can adjust treatment or make decisions to close the recreational beaches in Ohio or not. So after we have all of our field, laboratory, and environmental data, such as weather data, lake level change, things like that, we put that all into Virtual Beach. And then also the cyano chlorophyll A estimates from Cyan website. Those cells per mil will go into the model as well. We can build 2 types of models, a more like real-time model that factors are easily continuously measured. You don't have to wait on a sample to be analyzed in a lab, or a comprehensive model that has everything into it. And then, so I don't have time to share a bunch of models, but I did pick one. And then this is just an obligatory slide that these models are still in review. So this is the Cedar Creek Lake model, the comprehensive model with the satellite estimate in cells per mil. And this is predicting a 1 microgram per liter threshold exceedance for microcystin, and the other variables in the model used to predict that microcystin level ammonia, dissolved oxygen, and rain 2-day sum. And then in Virtual Beach, the software, you go through all the modeling steps, you analyze your data. So sometimes your data has to be transformed because that environmental data isn't normally distributed. So we go through all these model evaluation criteria. So this model was developed by using AICC, which is corrected for a little bit smaller data set because so far we only have 40 discrete samples in the model. But we had an R squared of 0.68, and all the variables had a p-value of less than 0.05. And then another thing we like to look at USGS is the accuracy, specificity, and sensitivity. And we usually looking for greater than 80%. And that's building a robust model. And then in this graph, you can see this is another output from Virtual Beach, where you can see your correct exceedances. So the model's predicting correctly. In the bottom right, false negatives. This one didn't have any. It had a few false positives, which are in the top left there. And since we are kind of erring on the side of caution and wanting to protect human health, we say a few false positives is okay if we go to use these models. And then, of course, the correct non-exceedances are below that. So just another visual way to see how your model's performing. And then what exactly can we do with this data? Well, the models, again, can be used for real-time swimming advisories. They can be used by water treatment plant managers to adjust their drinking water treatment to the current conditions. We can identify the specific factors related to cyanotoxin presence at your specific site or lake. And then the satellite cyanobacteria chlorophyll A estimates incorporated into the models are a pretty promising tool and can help augment the cyanin early warning system specifically for toxic HABs. And then real quick, these are just acknowledgements for all the local and national advisors and cooperators with the project. And then with that, I'll take any questions.
Okay, great. We got about 10 minutes for questions overall. Do we have anything specific for Jessica? Nope. I got one. I was just wondering, with the model that you showed at the end, are all those factors from like continuous monitors, like the ammonia and things like that? Or are you taking the cyan data and then like plugging those in based on lab stuff?
So that specific ammonia was a lab concentration. So the sound we have it doesn't have ammonia on there. It had like water temp, DO, pH. We had BGA on there as well. But yes, so some of them are lab concentrations, and then some of them are from the sonde, or like the weather data I pulled from publicly available sites. We also had lake level change for that lake as well. So I combined all those things. And then Virtual Beach will, since it's multiple linear regression, it will come up with the best fits models. And it gives you a top model for predicting that independent variable, which is microcystin.
All right, thanks. Do you have anything online, Pam?
Sorry, I lost my mouse. I don't. We don't have anything online. Oh, no, we do. I'm sorry. Excuse me. Steve Hansen asks, for building these models, what frequency of toxin detection would you think is necessary?
That's a really good question. I would say probably at least 20 to 30%. And that might give you in the software Virtual Beach, it might, if from year to year, if you have different conditions, maybe the next year you don't have that many positive hits, it can end up changing your model from the year you started to the year you kind of ended sampling. So it's pretty important to keep in mind how those different number of samples, if they're positive or not, help you build that robust model.
Great. Thank you, Jessica.
Okay, great. We got just about 5 more minutes left in the session. Do you have a question?
Just to follow up to that, then kind of going back to the sites where you were collecting your information to help drive the model. As you know, these lakes are quite variable, and there's heterogeneity of the cyanobacteria, pileups close to the beaches, et cetera, versus some of the open water sites. So I'm just wondering how do you strategically place your monitoring sites to account for this variability within the lakes? Obviously the cyanocytes can't be next to shore. I mean, there's the contamination or shallow waters which would contaminate the image. But do you take samples close to shore, close to the beaches, as well as out in the open waters to help strengthen these models?
Yeah, that's a very good question. So that's something we were probably going to look at in the future. This first part was just seeing if we can incorporate that satellite imagery data into models. But in our previous projects or previous models, we did pick like near shore sites, like specifically if you're worried about closing down the beach. Now at Cedar Creek Lake, on the opposite side of the lake is the water treatment intake plant. So we could also, sample there as well. But this first initial part of the project and the funding we have so far was just kind of, we're covering the expanse of the lake and having to make sure that those sites are away from the shore because you do have the mixed pixel issue.
Okay, any other questions for Jessica or for the whole panel while we still got a few minutes? Nope. You got anything, Pam? Oh, we got one here. Okay.
I have a question for the folks that briefly mentioned a little bit about hyperspectral work. So I think it was like Reagan, Laura, Bridget, a little bit about hyperspectral. I'm curious what you guys think is what kind of information you can get from the hyperspectral information that's new in terms of what are you hoping you can do to advance maybe looking at freshwater harmful algal blooms? Do you think you can actually get to the point of seeing if a more toxic taxa is present over another one? Or I'm just kind of curious where that might be going.
I'll start just quickly from our perspective. Since we're looking at the hyperspectral microscope as well in one of the projects, part of that is going to look at the kind of life cycle and not only toxin producing, but where in the life cycle those organisms may be. And so that's pretty early on, but part of what we'll be putting together in what we're viewing. So right now we're looking at various pure cultures and then also the real world matrix and understanding if that scope can differentiate between the two and at a lab bench scale level start to do more investigative applications with that. So that's from that side, but I'll open it up for the satellite piece too.
I don't know if Bridget, when she's getting up and going, one of the ways that we look at the hyperspectral and utilize it is actually looking at community dynamics, in that the blooms aren't just all cyanobacteria, right? There you got a lot of diatoms in there as well. The hyperspectral really allows us to get at that competition level so that we can see, based off of all the other information that we're getting out of our buoys and our water monitoring, how different species or different groups of phytoplankton are using the water and what triggers maybe cyanos winning versus the diatoms winning. And then that can help us better just predict what's going to happen, not only now, but in the future, but also how we might be pulling levers in the way we manage the water system. So is it just nitrogen? Is it just phosphorus that we should be looking at? Should we also be considering pH? And we can answer those types of questions with the help of the hyperspectral, because it takes lots and lots of time to do counts. Hyperspectral is a lot quicker at getting us that information. And Bridget.
Yeah, no, that was perfect. Yeah, just that species separation with the Cyan project. We do have like a little double check to see if something's cyano or not, but yeah, the bands are limited and we do kind of sometimes throw in non-cyanos into that or vice versa. So should help clear that up. So perfect.
Okay, any last questions? Do you have something to add, Matt?
Yeah, I just had a reference to offer you. There was a 2023 paper by Carl Legleiter called, it was on SMASH, which is the spectral mixture analysis for surveillance of HAB. So yeah, if you're aware of that, I thought that work was really cool and really in line with exactly what you were asking. So.
Yeah, and just a plug for Carl's work. I think that they are always looking for collaborators to collect samples to line up with some of that satellite imagery to refine that model. He works for the USGS.
Okay, it's about 10 o'clock. So any last questions, either in person or online?
We have one online here from Alexis Fisher regarding drones with the hyperspectral sensors. Approximately how much do these systems cost? And can you recommend the gold standard model? I'm wondering if something like this would make sense for a municipality.
So it all also depends on what type of sensor that you put on. So one of the things that we are designing and testing this year is a point source drone with a hyperspectral. Well, it's not hyperspectral then; it's just a single point spectrum that can be purchased all under 10K. So that's much more reasonable for some of the municipalities. The system that we've tested previously in the past has been closer to like, I think the sensor itself was roughly 40K and then the drone was 60K. And some of that is barriers within the federal government because we can purchase US based products. So I will mention that you could probably get cheaper and different products that aren't necessarily within that cost band.
All right. I think we're all done. If you want to wrap up, Pam.
Sure. I just want to say thank you for everyone for attending, and thank you to our incredible group of federal, local, and you know, all the scientists are on here. If you would like to get in touch with them, their contact information is in the WOBA app, or you can reach out to Kyle and I. I work for the USGS, Pam Riley, and Kyle works for the Water Supply Authority in New Jersey. And we just want to say thank you very much for everyone for attending today.
Thanks, Pam and Kyle.
All right. Thank you very much. And thanks so much to the tech team here too.
Yeah, thanks to the tech team. We made it work, everybody. Super excited.